Hello, everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. In this series of 4 talks, we're looking at phylogenetic opportunities and challenges from bacteria and archaea. Our first talk was from Laura Hug about recent discoveries of new groups of organisms. Our talk today will be from Xavier Didelot, and next we'll be hearing from Ten Vaughn. And we've just added one new talk. It's not up on the website yet, but it's going to be from Ford Doolittle. It's going to be some sort of philosophical thing. I'm not sure exactly what. So that should be very fun. I wanted to remind people that the Q&A app is gone now, so you can ask questions through Twitter or IRC, as described in the attending section of the Phyloseminar website. As I mentioned, today's speaker is Xavier Didelot. Xavier did his doctorate in statistical genetics in the Department of Statistics at the University of Oxford. He then did a postdoc in the Department of Statistics at the University of Warwick. Then in 2010, he moved back to University of Oxford as a leadership fellow in genomic microbiology before taking his current position with Imperial College London in April 2012. Welcome, Xavier, and thanks for participating.
All right, thank you very much for the introduction, Eric. It's a pleasure to be here. I'm Xavier Didelot, speaking here from Imperial College London. And I'm going to start sharing my slides with you now. That's it, you should be able to see that, right. And so my talk today is entitled Modeling Recombination in Prokaryote Phylogenetics. I mean, really by that I mean mostly bacteria, really, but to sort of fit with the theme of this lecture series, I've sort of broadened it to prokaryotes. So there will be one example of archaea, but really mostly it's bacteria that I have in mind. And so the first thing we should know about prokaryotes, if we want to study their population genetics, is that they reproduce clonally, right? So if we start with a single bacterial cell or prokaryotic cell here at the top, the way it reproduces is by copying its own DNA, and then the cell will sort of get bigger and bigger and split into 2 halves, and each of the two daughter cells will have one copy of the DNA of the whole genome, right? So each of those 2 cells has a single parent, which is the cell at the top. Okay, and what does that mean in terms of population? Well, one way to think about it is as a Wright-Fisher population, where we have several generations. That's a really well-known process. I'm just going to talk about it quickly. Several non-overlapping generations in the population, all of them of the same size. So in this case, we've got a population size of 8 individuals. And this is the first generation here at the top. And each generation is non-overlapping, right? This is the first generation at the top, then the second one, third one, etc. And each generation is formed by uniformly sampling with replacement from the previous one. So we get this type of graphs where we can see each individual has a single parent from the previous generation as implied by the clonality reproducing. And so if we run this process for a number of generations like that, and we stop and we look sort of back in time at we take a sample from the sort of latest generation of our population here, sample of size 4, and we sort of trace back in time its ancestry. What we get, of course, is a tree looking like this, and we can forget about everything else that is not ancestral to our sample of size 4 here, and we end up with a tree looking like this, right? Nice and simple. Familiar with the Wright-Fisher model, of course, knows that if you have a Wright-Fisher population or a number of the models, in fact, and you think about the genealogy back in time, that is described by the coalescent model, right, which is represented here. So the coalescent model describes the genealogy back in time of our sample of size 4 in this case, and it's a pure death process with rate k choose 2, where k is the number of lineages currently living, right? So if we start at the bottom here of the screen, we have to wait an amount of time which is distributed with k choose 2 here until one of the until 2 of the 4 lineages coalesce, right? And we end up with just 3 ancestors, and then the process is repeated with 3 ancestors down to 2, and then from 2 ancestors down to 1. Okay, so that's the basic genealogical process we get if we don't have any recombination. And for some species, such as Mycobacterium tuberculosis, where there's no or very little recombination, that would be pretty much the end of the story. But for most prokaryotes, there is in fact some level of recombination that is going to make the ancestral process a little bit more interesting. For some bacterial species, such as Staphylococcus, perhaps, for example, there's only a moderate amount of recombination, so the evolution can be thought of as a tree with added recombination. And for some of the species, for example, Helicobacter pylori, there's quite a lot more recombination, so that so tree-like evolution becomes almost completely irrelevant. So how can we fit this recombination process into our ancestral process? Is the next thing we're going to ask ourselves. So firstly, I should say there are 3 routes to recombination in bacteria. So starting on the left with the donor cell, the first thing that can happen is that this donor cell dies and its DNA is released in the environment. And there's this process called transformation, whereby a recipient cell will pick up one of the bits of DNA, and that bit of DNA might become integrated through recombination into the recipient genome. Second process that can lead to recombination is called transduction. That's when a phage is going to act as a vector. So instead of encapsulating its own DNA, the phage by mistake will encapsulate a bit of chromosomal DNA and will go and inject that into its next infectee, the recipient cell in this case, and it's possible then that that bit of DNA will become integrated into the genome through recombination. And finally, the third process is called conjugation. That's when the 2 donor and recipient cell come into contact with one another, and then there's potential for exchange of DNA and then recombination into the chromosome. So those 3 processes are well described. I won't actually be talking much about those 3 processes. In each case, we will be summarizing the net effect of those 3 processes as recombination. Right. And so what can happen when you have recombination? There's quite a lot of things that can happen. We might have some new genes being inserted into the genome. We might have some previously existing gene being erased, being deleted because they are being copied on top of, or they are being removed by recombination. We can have replacement of an allele of a gene by another. We can have replacement of only a part of a gene, perhaps central part or any part of the gene. Or we can have a mixture of these different effects with a mixture of homologous replacement and non-homologous insertion. But here, for the purpose of this talk, I'm going to be focusing mostly on homologous recombination, right? So I'm going to be asking myself, what is the effect of recombination on the ancestral process when we look at the parts of the genome that are conserved across a sample of prokaryotes? Right, so we're not so interested in this process of insertion and deletion of genes, which is fairly obvious to spot. We're looking purely at the regions that are being consistently found in all the bacteria, and what is the effect of recombination on them. And so the effect on the ancestral process is in fact well known, has been described for some time, and what we end up with is not a tree anymore, but a graph, also known as the ancestral recombination graph, or ARG. And so what this is a birth-death process instead of a pure death process, where we now have coalescence happening still at rate k choose 2, but we also have recombination happening at a rate rho times k over 2. So what that represents is there's a certain rate of recombination rho over 2 that can happen for each of the k lineages living at a given time. And so the compound rate of recombination for the k lineages is rho k over 2. So we end up with a graph looking like this. We still have some coalescent event happening when 2 lines of ancestry coalesce and become 1. But we also have some events like this one, where 2 lines where a single line of ancestry splits into 2 lines through the recombination process, and that's an example of a birth looking sort of back in time, and so recombination creating 2 lines that we now have to follow the ancestry of. Okay, so when you see, there's a lot of literature, of course, on ancestral recombination graphs, going back to Hudson in 1983, I believe. And the ARG, when you find it in literature, usually the ARG that is being described is the ARG with crossing over, which means that when recombination occurs, a point is chosen uniformly at random on the gene or genome, and the material on the left will follow one line of descent, and the material on the right will follow the other line of descent. So if we look at the at this event here at the bottom in the middle, we've got the ancestry of the genome. The fragment on the left of the midpoint follows the line on the left, and the fragment on the right follows the line on the right. And same thing for the other reconstruction events. So there's symmetry in the role played by the two parents because they contribute, they are expected to contribute roughly the same amount of material if the point is anywhere along the genome. And like I've said, this is a version found in the literature. When people talk about ARG, usually this is the process that they mean, right? But for us interested in prokaryotes and bacteria in particular, what we want is a version of the ARG that is slightly different because we want the ARG with gene conversion, right? And what that means is that when recombination happens, instead of having the two parents contributing equally, what we have is that there's a small fragment, maybe of mean delta. We're going to use delta for the average length of this recombined fragment. That a small fragment of the genome is contributed by the donor cell, and the rest is contributed by the recipient cell, which fits with what the three processes of recombination I described earlier, where there was a donor and a recipient cell, right? And that changes the process quite radically because now we have asymmetry between the two parents, the recipient and the donor. The recipient contributes a lot more, and the donor only contributes this small fragment. Maybe it's even a very short fragment. It could be just a few base pairs, really. And this is a version of the ARG that is relevant for prokaryotes because the recombination process acts in this way with the donor contributing only a small fragment, right? And this version of the ARG, so it's also a well-described version called so the ARG with gene conversion. For the rest of this talk, when I talk about ARG, this is the version that I mean because that's the one that's relevant to prokaryotes, and it was first described really in detail by Ruth and Hein in their paper in 2000 that you have the reference on the screen. Okay, so this is the ancestral process that we are interested in to describe what we expect the ancestry of a sample to be in a bacterial population under some amount of recombination. And if we want to simulate this process, so simulating the ARG with gene conversion, that is relatively easy. There's a number of softwares that have been published to do that, starting with, a lot of the standard software can do it, such as MS, for example, can simulate gene conversion on top of the crossing over process. But specifically designed for prokaryotes, we've got a number of software as well. So SimMLST, for example, will simulate this process. SimBac, more recently, published last year, will simulate this process. And the only difference really between SimMLST and SimBac, it's the same process, but SimBac is optimized because it has a more clever way of following the line of simulating the recombination event. Instead of SimMLST, it simulates a lot more recombination event than it needs to because it's not calculating the exact probability that a recombination event actually has an effect, whereas SimBac does. Even more recently, a paper has been published, has been submitted to bioRxiv called FastSimBac, which actually simulates from a slightly different process that is a very small approximation to the full ARG process, and by doing that, you can simulate a lot faster. So simulation, really, once you understand the process, is relatively easy and can be done fairly quickly. And if we have a way to simulate ARGs with gene conversion, that means straight away we can use the simulation to infer some main parameters from a data set. So we can use approximate Bayesian computation to basically simulate a lot of data sets, compare them with what we have, and narrow down at least the field of possibilities for what the parameters might be implied in our real data. But this is not really full inference under the ARG. It's just ABC inference, so not based on the full likelihood, not based on the whole process, and not inferring all the parameters. That's only going to work to infer the main parameters, perhaps the recombination rate or the length of the recombination events delta. If we want to do full inference, and by that I mean reconstruct all the ancestral process, the whole ARG, the whole ancestral graph that has led to the data set that we are looking at, that's a much more complicated process, really, and we need to, and trying to actually do this inference under the full ARG with gene conversion process that I have described is a notoriously difficult problem, and basically can't be done for data sets of a large size like the one we are interested in, right? I mean, I'm talking data sets of the order of a few hundreds to perhaps even a few thousands whole genomes of bacteria, which is what current sequencing technology allows us to generate, and doing full inference under the ARG is not going to work for such large data sets. So what we need really is some good approximation to the ARG process that lets us do inference under this approximation and still bears enough resemblance with the ARG process that it is informative about what actually happened. Okay, and so when you think about, again, the role of recombination in phylogenetics, so I've said this already, recombination is a great challenge to phylogenetics inference. So that's true both of the ARG with crossing over version as well as the ARG with gene conversion. And really what it does, its effect, one way to think about it is that it causes the ancestry to be a graph-resonant tree, we've said that already, but also it causes different parts of the genome to follow different trees, right? So if we have this ARG shown here, which is the one we had in the previous slide, and if I follow, if I focus on the leftmost point of my genome and I trace the ancestry of that site, I actually get a tree that is embedded in my graph, which represents, if you want, the local tree for the first site. Now, if you do the same thing on the right-hand side with the last site, we find again a tree embedded in the full ARG that represents the local tree for this site, but it's not the same tree as the one we had for the first site because the two sites have been affected in different ways by the recombination events. So a lot of algorithms looking at ARG with crossing over try to exploit this feature and look at how the tree changes as you go along the genome. But in the special case of the gene conversion ARG, we have one additional concept that is going to be very useful to use as the basis of our approximation, and that's the clonal genealogy concept. And that's a concept that doesn't exist at all with the ARG with crossing over, that is really specific to the ARG with gene conversion. So the clonal genealogy, let's say what the definition is. It's a tree that you obtain. So from the ARG, if you always follow the line of the recipient of each recombination event, you end up with a tree that is the clonal genealogy. So you can see that's what we've illustrated here on the right-hand side, again, using the same ARG as in previous slides. For the first recombination event in the middle, I'm following the line on the right because you can see that the one that is that of the recipient, the one, because the one on the left clearly only contributes a small fragment, that's the donor. And likewise, for the recombination at the right-hand side, we're following also the line on the right, and we end up with this clonal genealogy. So if there are very few small recombination events, you can see that many of the sites will in fact follow this tree, the clonal genealogy. But if we have more recombination events, it's quite possible, in fact, that none of the sites follow this clonal genealogy. If you did the process we were doing in the previous slide for every site one by one, you might not have a single one for which you end up with this clonal genealogy defined by following always the line of the recipients. And yet, even in that case, this tree does exist as a concept. It's a tree that is embedded in the ARG, and it is quite central to the ARG. It forms the basis for what we expect to find in common to a large extent between the different sites. Even if none of the sites really follows that genealogy, we expect many of the sites to look to some extent like it, because for a site on average, there's more chance that we follow the line of the recipient than the site of the donor, just because the donor contributes less DNA. So it really is a tree of central interest for inference, and it's going to be really useful to use it as a basis of inference and at the basis of some of the approximations we need to introduce to do inference. Okay, so one of the first ideas we could suggest to approximate inference would be to just ignore recombination altogether, right? We could say maybe we're going to apply a standard phylogenetic method that doesn't model recombination, and we're going to hope that perhaps the tree that we get with this method is going to be approximately equal to our clonal genealogy. Okay, and that's a method that kind of makes sense. So it's saying that basically our tree model is an approximation for our is the approximation that we're going to use for our full ARG model. And we can see clearly that's going to work quite well if we don't have too much recombination. But the more recombination we have, because in that case, if we don't have much recombination, a lot of the sites will, in fact, have for local tree the same tree as the clonal genealogy, and so will be informative directly about this clonal genealogy. The limit would be this case where we have no recombination that we talked about a bit at the beginning. In that case, obviously, we end up with this approximation being working really well. But what happens as we have more and more recombination? Well, this is what was put to the test here in this paper by Jess Hedge and Danny Wilson. And what they found is that as you increase the recombination rate, of course, the accuracy of the different phylogenetic algorithms without recombination goes down. It's not too bad in terms of the topology of the tree that we get. Even at a fairly high rate of recombination, we still have many of the branching that are correct, but the branch lengths are going to be really badly affected. And that's what is shown here. If you try to deduce from the branch lengths what was the growth rate of the population, you end up really overestimating the growth rate, and that's because a lot of the trees will be affected by recombination becoming more and more star-like, and those star-like trees, if you're not aware that there's recombination, they look just like the type of tree you would get under a model with exponential growth of the population. So what the plot on the right shows us is that as you increase the recombination rate, the trees become more and more star-like, and it becomes impossible to estimate, and the branch lengths are becoming really wrong and easily misinterpretable. So ignoring recombination doesn't work fantastically, of course. I mean, it has the advantage to be simple. But perhaps what's even more worrying, even if it worked well, even if we did get the clonal genealogy right, the clonal genealogy is not the only thing we want to learn about often. We want to learn about the recombination process itself very often. So ignoring it altogether is not going to help us with that. I mean, recombination is involved in a lot of things, antibiotic resistance especially. For example, we know that resistance often happens, recombination is often involved, and so we won't be learning anything about it if we just ignore it altogether. So this next model, the ClonalFrame model, tried to go one step further, really, from just ignoring recombination to integrate the recombination process, or a very simplified version of the recombination process, into a phylogenetic framework. And so it is based on a tree, which is the clonal genealogy tree, this tree that you would get if we only follow the line of the recipient all the time. And on top of this tree, we have mutation events happening at a rate θ/2, just like in a standard phylogenetic model. But we're also going to have a recombination process happening at rate ρ/2. And when we have recombination happening, for example, the little red arrow here, what happens then is, so normally in a normal ARG, we would have to follow the line of that donor of the recipient, but here we won't be doing that in the ClonalFrame model. All we say is that firstly, we determine which region of the genome is being affected by the recombination. So we have a starting point that is uniformly distributed along the genome, and the length of the recombination, which is geometrically distributed with parameter delta. We've said this before. Delta is our parameter for the length, the average length of the recombination events. What happens to the recombined region, this little region in red, in all of the daughters from the event? Well, we're just going to say that in those regions, there's a high rate at which substitutions are being introduced, and that's the rate nu here. And all those little, in the little red regions, you can see we have those little red crosses that have been introduced and that are being passed on from generation to generation. The idea being that if the recombination came from quite far away, in this recombined region, we're going to see a lot of polymorphism being brought in, and that's going to be the effect of recombination in this ClonalFrame model. So it's based on an idea of a recombination coming from quite a distant source. But this parameter nu, of course, determines how far the source is. If nu is small, then we have less recombination less polymorphism being introduced by recombination. Okay, so that's the basis of the ClonalFrame model. And there are two implementations of this model. The first implementation, which is the original one back in 2007, we used the Monte Carlo Markov chain with Daniel Feluche, my PhD supervisor at the time. We designed a Monte Carlo Markov chain to sample from the space of ClonalFrame models. And this version really was designed mostly for MLST data, because that's what the type of data that was predominantly available at the time. This version is no longer maintained. Instead, I would encourage anyone who wants to use ClonalFrame to use the new version, which is ClonalFrame ML, that we devised together with Danny Wilson. And you have the reference here on the slide. So it's not Bayesian anymore. It's not using an MCMC anymore. Instead, we switched to maximum likelihood techniques, which are faster. And as a result of that, based on exactly the same model as the one I described, the ClonalFrame model, we can handle much larger data sets as before. We can handle hundreds of whole genomes in a matter of hours, really, in terms of computational time. So to show you an example of what the output looks like when we use ClonalFrame ML, we've got here on the left, this is ClonalFrame applied to about 100 genomes of MRSA from the whole species MRSA. We've got the tree corrected for recombination shown here on the left, which approximates the clonal genealogy, right? And on top of that now, unlike what we were doing before when we were just ignoring recombination, we've also learned something about recombination, and we can look at the heat map on the right that indicates for each of the branches of the tree, each row of the matrix, it tells us where recombination events have happened, which are shown by the little blue, the dark blue dots and bars. And that's the way you can read those plots from ClonalFrame 1 and ClonalFrame ML. Okay, so that's for the application of ClonalFrame. So really, I'm not so interested in application here. I want to focus on the models and how to build better models. And so if we think about the ClonalFrame model and what how it works and what it represents, we have this, like I've said, the effect of recombination is really as if the source was external. And that's not just because I'm assuming nu is high. It's mostly because I'm introducing some new polymorphism with each recombination coming in. And I'm not copying polymorphism from one part of the tree to another as the normal ARG would be doing. No, it's a much more approximate process than that, really. So how does that work if the recombination was in fact internal to the data set? Well, some of the events will be captured, mostly the one that was a lot of polymorphism has been brought in from one place to another, and some events won't be captured that well. Okay, so we only partially capture within population recombination events. And sometimes that's a problem because we don't do very well at detecting the full extent of recombination that happened. And also the other limitation clearly is that if we want to learn about the origins of recombination, then ClonalFrame itself doesn't tell us anything about that because it doesn't try to trace the ancestry of the imports, the recombination imports. So if we want really to know something about that, we can do it a little bit. So in this paper, for example, the example shown here is from a dataset in Salmonella, and what we did that for each ClonalFrame event that was inferred, we compared that with we compared the polymorphism introduced with what is found elsewhere on the tree, and we were able to post-process the output from ClonalFrame and have a good guess at where it might have come from, and to reconstruct this graph, which shows us for each for different lineages of Salmonella how many events of recombinations were coming from one lineage into another, or staying within one lineage, or coming from yet another source, which is the external node at the top. But so that's not really satisfying, this method of doing it as a post-processing step. And even if it worked, even if it works well for those events that have been detected, clearly a lot of recombination events will not be detected because the ClonalFrame model, the recombination process is being so roughly approximated by when we say that new polymorphism is being introduced rather than carried from one part of the tree to another. And so, yes, hello?
Oh, it sounds like maybe you're going to describe, okay.
Okay, yes.
It's going to give the better model. Okay, go ahead. Sorry.
Absolutely, yes. So in fact, this slide was really just just there to bridge between ClonalFrame and ClonalOrigin. So try to motivate why we're not happy with the ClonalFrame model completely, and why we want to build a better model. That's exactly what the ClonalOrigin model tries to do. But let's not throw away ClonalFrame altogether yet, because ClonalFrame, it's easy to work with because it's a tree basically. It's not really a graph. It's not an ARG. It's just a tree where we've added a non-graphical process of recombination, and that's why it's easy to work with and to calculate likelihoods and do stuff like that. So really, what we want, trying to build a better model, is to keep this nice feature of ClonalFrame, keep the clonal genealogy, which is central, and just improve the recombination process around it. And so that's exactly the idea of ClonalOrigin. That's how we build this model. We are now going to try and model the origin of each recombination events, something that we didn't do with ClonalFrame. But we're not going to reconstruct its full ancestry for each import, each recombination. All we're going to do is say that each recombination event has an origin, which is a point on the genealogy. So I'm going to show an example of that in a second. But maybe I should show you the example now. So this is an illustration of the ClonalOrigin model. Now you can see it looks like a graph, really. It looks a bit like the ARG graphs we had before, but really it's not. What it really is this tree process, this coalescent process, which is shown in bold, with a couple of edges added, which are the red and the blue edge here, which represent the recombination process happening on top of the genealogy. And you can see that each of these recombination events has an origin, which is noted here, A1 and A2, has a place where it arrives, which is the points B1 and B2, and it affects a given segment, which is X to Y, X1 to Y1, and X2 to Y2. And we can write down very simply the probability of such a graph, this ClonalOrigin graph, because we can decompose it between the part that is the clonal genealogy, which is simply given by, so this probability of T is simply the probability of a coalescent tree. And we can write what is the probability of the recombinant edges on top of that tree, very simply as the process of those, as a product for all of the recombinant edge of the probability that event had the given origin, the given destination, and stretch from X to Y. So we can write the prior probability of a clonal origin graph. And in terms of calculating the likelihood, something we want to do, of course, if we want to do inference, we can deduce from the graph what is the local tree at each of the sites along the genome. And then we can use something like Felsenstein pruning to calculate the likelihood at each site given its local tree as defined by the graph. So if we think about this ClonalOrigin model, and we think about, so clearly it's much more similar to the ARG model that we had started with, but there are still two key differences really from the ARG model. And the first one is that we are not completely following the ancestry of the recombinant edges. Instead of that, we don't allow those lines, the red line and the blue line, to recombine again. We just regraph them somewhere above the tree. So it's like saying that recombinant lines are not allowed to recombine again, which is kind of justifiable because they don't carry a lot of ancestral material. So there's the probability that they recombine effectively is quite small. And the second approximation we're making is that those lines, the recombinant lines, they just regraphed somewhere with the clonal genealogy. They are not allowed to coalesce with one another as would normally be allowed under the full ARG model. But obviously, working with such a graph would be a lot more complicated if we allowed it. So because then the edges wouldn't be independent from each other, which is the nice feature we have here at the moment. Okay, so we implemented this model back in 2010, and so we did some comparison with, between ClonalFrame and ClonalOrigin, and as we would expect, really, we find that so if we simulate under the full ARG with gene conversion model, ClonalOrigin does a much better job at inferring the real recombination rate rho compared to ClonalFrame, which has a tendency to miss a lot of the events because, like I've said before, ClonalFrame will only detect the events that have had a significant impact. It will miss a lot of the events and therefore underestimate the recombination rate rho. And there's a lot of nice things we can do with this ClonalOrigin model, which in fact, even if we were able to infer the full ARG, it wouldn't be so easy to do with. To do. I mean, the ClonalOrigin model, it's an approximation that makes it easy to represent things. Sorry, it makes it easy to infer the graph much easier than the full ARG, but also makes it easy to represent and interpret those recombination events. So here's an example of that where we applied ClonalOrigin to this data set of Bacillus cereus genomes, and we can browse along the genome. So we've got our clonal genealogy here on the left, which represents has the 13 genomes here are clonally related with one another. And we can browse along the genome to see what recombination events have happened in different genes. And for each recombination, so we've got this little plot represents the height, represents the probability of recombination, and the color represents the origin of the recombination events as shown on the tree on the left. So where we have, for example, a green blob, right, for gene number 3 in gene BC3152. I don't think you can see my cursor, but that's it's genome 3 and gene BC3152. And that means that there's been an import for that. There's been a clear recombination event for that genome of that gene coming from the genome that is of that light shade of green, which I believe is genome 10 or its direct node directly above it. And so that's quite a nice way really to represent what recombination events have happened along the genome. And if we are interested in a given gene and we want to see how it differs from the clonal genealogy, we can do it quite nicely in this way. And so building those plots is really quite informative. It's a nice way to show what events have happened, as opposed to if we were trying to reconstruct the full ARG. Even if it was possible to do it, it would be quite difficult really to summarize. And the ClonalOrigin model makes things much easier to summarize, to interpret, and to display. So in terms of implementations, the original implementation in the 2010 paper used reversible jump Monte Carlo Markov chains. That's the reversible jump bits come from the fact that we have recombination edges being added and removed. So we work with a parameter space that has a non-fixed dimension. So we need to use reversible jumps. And at the time, we put a lot of effort into trying to infer both recombination events and the clonal genealogy jointly, but it didn't work really well, at least not for the size of data that we wanted to deal with. So we ended up inferring just the recombination events, assuming prior knowledge of the clonal genealogy, which this clonal genealogy might come from, for example, from ClonalFrame. So we were using the conditional independence of genomic regions to scale to whole genomes because we're able to work out for different regions quite far on the genome, along the genome, what the recombination events were in those different regions. So more recently, a new implementation has just arrived called bacterial that was devised by Tim Vaughan and colleagues, and that version is implemented within the BEAST2 framework. It jointly infers the clonal genealogy and the recombination events, and I won't be saying more about it because Tim will be describing it next week, so I'm looking forward to that. Okay, so coming back to the original ClonalOrigin, so those two, just to be clear, those two implementations implement the same model, so which is a ClonalOrigin model, and so coming back to the results we got with ClonalOrigin, the software. One of the most interesting features, I think, was, so this is the very first application we did to this data set of 13 Bacillus genomes. And you can see that there's, what this plot represents is the probability of recombination coming from one part of the tree into another part of the tree, so with origin being on the x-axis and destination being on the y-axis. And it's being scaled against the expectation for how many events we expect to see under the sort of standard ClonalOrigin model, where we have edges happening as I've described. And what we find that there's a discrepancy here between the model and what we infer, because there's clearly two, if not three parts to this matrix, one which is much brighter. So the first few genomes have been recombining a lot with one another, and the last few genomes have been recombining a lot with one another, but there's not so much recombination happening between the first genomes and the last 3 or 4. And that's a pattern we saw repetitively in several examples. So here's the example of archaea that I promised at the start. There would be an example from archaea. So this is work we did with Raquel Whittaker on Sulfolobus islandicus. And we find again that there were, so we applied ClonalOrigin, and we find that there were at least 2 groups, the blue groups and the red groups. And the blue group has been recombining a lot with one another, as you can see with the yellow cells, and the red group has been recombining quite a lot with one another as well, but there's been little recombination between the blue and the red groups, as shown by the blue cells. And just to show you a third example, here is an example, a slightly larger example where we were analyzing 27 genomes of E. coli, on those 27 of E. coli, we see a very similar pattern where we've got the A and B1 groups that have recombined quite a lot with one another, and especially within A and within B1. We have B2 that has recombined quite a lot, again, with itself, but little recombination, less than expected recombination coming from A into B2, for example, or from A into E or E into B1, etc. And we can represent that in a graphical way in the plot on the right, which shows for each group coming from one group into another, the number of recombination compared with the expected number of recombination. And we find there's always more recombination going on within groups or between closely related groups, as opposed to recombination happening from one group to another or between sort of distantly related groups. And so what that represents really is this tendency of recombination that we see in all those data sets for recombination happening more between things that are closely related. And that's not something that is encapsulated in the ClonalOrigin model. In the original ClonalOrigin model, if we have a tree like the one shown here, and if we consider 3 recombination events shown here in blue, green, and red, they all have the same probability of happening, because they all arrived to the same place and they all came from the same trench in time, from the points A1, A2, and A3, which makes them equally likely under the ClonalOrigin model. Or under even under the full ARG model, they would be equally likely as well. So what we did was to go one step further, or to explore a refinement of ClonalOrigin, if you want. And this is a paper we did with Azim Ansari. And we added an extra term to the probability of those recombination events that represents this tendency for recombination to happen more when there's less distance between things. So here in this example, the red event is going to be now more likely to happen than the green event, which itself is more likely to happen than the blue event. And exactly how much more likely is going to depend on the distance between the points, arrival and departure of the recombination events, but also on a parameter lambda, which represents this tendency for recombination to be biased in this way. And what we showed in this paper is that we were able to infer the parameter lambda, so this tendency for recombination to happen more between things that are closely related. We were able to infer this parameter based on the same data set of Bacillus cereus that I've shown before. And why it's important is this tendency of recombination to happen between things that are closely related is exactly what can lead to speciation in bacteria, where well, it's one of several things that can lead to speciation. Really. So if we understand this bias in the recognition process, we are able to make predictions about when different groups of bacteria are becoming are going to recombine less and less and become more and more diverged and perhaps become separate species eventually. So that feeds into a debate about the definition of species in bacteria and about how speciation happens in bacteria. Okay, that takes me to the end of my talk. So just to recap briefly about what the different things I've been saying. So recombination is frequent in many bacterial and archaeal species, so we don't want to ignore it altogether. It's a process that often is interesting and is causing a lot of interesting things to happen, be it adaptation or resistance or etc. So it's a process that often is worth studying. But as soon as we incorporate recombination formally into the ancestry process, we end up with a graph, this ARG rather than a tree, which makes things much more difficult to work with in an inferential framework. So we need to devise some approximations to the ARG. The first approximation I've mentioned is the ClonalFrame model, which was the first attempt at incorporating recombination within a phylogenetic framework, but it didn't model the origin of events, which makes it quite approximate, really, and more suitable perhaps for when we believe a lot of recombination has come from external populations. And the second approximation I've mentioned is the ClonalOrigin model, where now recombination is modeled and has an origin, a well-defined origin on the clonal genealogy, but of course it's a more computationally demanding model to work with. And I believe there's still a lot of work to be done to try and exploit this model on large data sets. So modeling recombination is important, not just for better inference of the clonal genealogy, but also because we want to study this process for itself. And the example I gave you at the end on that, concerning sequence divergence, if we study the recombination process well, we are able to infer this bias in recombination and to say something about how speciation might ultimately happen in bacteria. And that's all I had to say. Thank you very much for your attention.
Thank you very much, Xavier. That was a very interesting talk. Obviously, a major body of work. Very impressive. I was sort of, well, first I should remind people that you can ask questions via IRC, which is described on the website under the attending section, or just tweet us at phyloseminar. But I'm afraid I'm going to start off by asking a really nerdy question, which is, I can kind of see how you would go about doing this in an MCMC framework. Of course, you have to do a reversible jump, and that's sort of annoying, but like it can be done, and you sort of sample recombination points and attachments, and maybe it'll be slow, but it seems sort of straightforward in principle. But optimization, like the ML optimizations, just sounds really hard. Like, how do you go about just like even starting to get a foothold about what should get connected to where, what should the boundaries be? Maybe once you have a rough idea, you can do a reasonable job of just optimizing locally, but how do you go about doing that?
So the maximum likelihood optimization in the ClonalOrigin model, which is what you were talking about when you're saying we're adding and removing some events, that's not something that we've actually done. When I was talking about maximum likelihood, that was in the ClonalFrame model. In the ClonalOrigin model, I think it could be interesting to see if we can perform maximum likelihood under the ClonalOrigin model. I would worry, like you do, that there will be a lot of local optimum and a lot of difficulty really mixing and jumping from finding the best optimum, and that might not even be really desirable, because then we will end up with. I don't believe there's an optimum that would necessarily be a lot higher than the other local maximum that we will get. So that could be attempted. The question would be: is there a good algorithm really to do it? Is there a good way computationally to do it? And like you said, it sounds tricky.
Okay. Sorry, I misunderstood there. I mean, I guess the other thing that I'm sort of curious about is, I mean, obviously viruses recombine too, and you must get this question a lot. I mean, is the point that you don't want to apply this sort of same framework to viruses because you're worried that maybe there's not as much of a backbone left over when the viruses are sort of recombining a lot?
So it's not clear, and I think it's going to be different between different viruses. If the process is best modeled as an ARG with crossing over, or an ARG with gene conversion, and really the algorithms I was describing are really focused on this latter problem. I think for HIV especially, which is probably the virus we know the most about its recombination process, as far as I understand, although I don't work on HIV myself, it's actually best represent. It actually works more as a crossing over process, in which case, in which case, the type of approximations I've been talking about are not especially appropriate, really. I'm not aware of anyone. I know some people work on viruses that wanted to apply ClonalFrame or ClonalOrigin at various points. I've never really seen any output from that, and I'm certainly not published output, and so it seems like it's probably not such a good idea.
Okay, we have a question from someone identifying themselves as Foo. So asks, what is the applicability of methods like these to deeper divergences, i.e. not trees within species, but among species or among classes?
I didn't catch all of that, I'm sorry. Can you copy me or quite? Okay.
I think the idea is that maybe some of the examples you showed were among reasonably closely related bacteria.
Yes, I was always working within a species, yes.
Yeah, so he or she is asking, what is the applicability to deeper divergence?
Okay, if we have a sample of bacteria that are from different species, I mean, if they are closely related species, it's probably fine. As we move sort of further and further and look at a sort of broader and broader spectrum of bacteria or prokaryotes, then the recombining process becomes quite different from the ones we've been discussing with the gene conversion ARG, because then this, if nothing, for one thing, this effect I was describing at the end of the bias in recombination towards recombination with things that are similar is going to become more and more acute, really, and we're going to see that more and more. But perhaps that's something we can account for, as we did in that paper. So that's probably something we would want to do if we were to try and exploit those methods. I mean, the methods I've described are really more aimed at looking at recombining within species. But if we were trying to apply them at a broad spectrum of species, we would probably want to use tools like that, yes, to model the bias in recombination, or perhaps to apply sort of bit by bit, really, to apply the model for each species separately and then collate the results in some way, including perhaps trying to work out when there's been an exchange from one species to another. That could perhaps be reconciled. If it's with a ClonalFrame model, it's fairly simple to do so because ClonalFrame doesn't assume anything about origins anyway. If it's with the ClonalOrigin model, then a recombination coming from a different species is usually ends up being a recombinant edge that regrafts above the root of the whole sample of the whole clonal GMV. So, and that's how we end up with polymorphism, a high level of polymorphism that is being represented by. So perhaps we could then post-process the output of ClonalOrigin being run on each species and look at those events where the departure point was above the root and try to see if that coincides and to what extent that can be reconciled with events coming from other species. Yeah. Again, I'm not aware of anyone really trying to do that, but yes, if someone's interested to do it, you can get in touch.
Yeah, sounds like people would be interested. I mean, so what is your next step? I mean, can you say a little bit about that?
Yes. So I mean, I think it's going to be really interesting to, and I've tried to give an introduction very, very briefly without spoiling anything for Tim Vaughan, who's talking next week. And I was very excited to see that people like Tim and his colleagues have been working on the ClonalOrigin model and trying to incorporate that in BEAST2, of course, which would make it, I mean, hugely more attractive for users to have it incorporated in a well-established phylogenetic framework as opposed to having to run a separate software, which doesn't have all the nice other features, of course, that BEAST2 has. I think, and I'm sure Tim will agree next week when you ask him, if you ask him, if we ask him, that there's still some additional. It's nice to see that people are still working on it, but there's probably still a lot that can be done. So the Bacter shares with ClonalOrigin the same way of proposing recombinant edges, which is to just sample them from the prior. So that means when we want to add a new recombination event in the reversible jump MCMC, we just randomly propose something coming from a random place on the tree to another random place. And of course, and that happened at a random place along the genome as well. And of course, the probability of proposing something correct by doing this random process is really quite low. So what really would make sense, I think, as a next step, would be to try and be a bit more clever in the way we propose the recombinant edges, looking perhaps at incompatibilities along the genome, those sites that those polymorphic sites that clearly are not compatible with the clonal genealogy as it stands, as it's being rebuilt or sampled in BEAST, and trying to look at introducing a recombinant edge specifically to try and explain those incompatibilities as opposed to just proposing them at random. So that would be a more complex proposal distribution to use, of course. It makes it more difficult both to devise and to say what is our proposal distribution. Of course, we need to know that to calculate the metropolis-lasting ratio, but also potentially would make it much faster to propose things that are that help explain the data and therefore to converge to the optimum of the ClonalOrigin model.
Yeah, what you described is really the crux, right? Calculating the proposal distribution, because I mean, you can come up with lots of different things, but I mean, are you going to be able to?
So I'm not saying I know how to do it, but I think it seems to me really there's something clever to be done about that, and often just from looking at the data, often you can sort of take a pretty good guess by hand or by eye at what seems to have happened. So if we could make the proposal distribution exploit these obvious features that happen sometimes in the data, then that would make them those algorithms more efficient, really.
Yeah, no, that's a good idea. I mean, and speaking of which, I mean, what is the current state of things for scaling? Like, what is the size of things you would feel comfortable recommending for people to run?
In terms of amount of data that can be dealt with. So ClonalFrame ML that we did with Danny Wilson 2 years ago now is really able to deal with large data sets, and I'm very pleased because the previous version of ClonalFrame had been designed for only MLST data and and of course there were more and more people trying to apply them to whole genomes, and I was getting more and more email of angry people unhappy about the performance and the algorithm struggling. So this new version, I'm really pleased that we were able to do this version, which can deal with much larger data sets and effectively really replaces completely the old version of ClonalFrame. And yes, so in terms of how much you can, I mean, I've successfully used that on 1,000 or 2,000 genomes, whole genomes of bacteria from a single species in a matter of maybe a day or two, maybe. So that really is quite fast to run. For the ClonalOrigin model, it's really I'm afraid it's more complicated, really, partly because of what I've said, that the proposal distribution are not clever enough. we end up with the ClonalOrigin original implementation, the one from the 2010 paper. We were already at the time interested to work with whole genomes, and so we have applied it. I think the larger data set we've applied it to is one of the ones I've shown with 27 genomes of E. coli. And the only reason why we can do that is, so firstly, because we don't try to infer the clonal genealogy, we just assume that we know it. Obviously, that's a bit bad. And secondly, we break down the genome into fragments that are effectively more or less independent along the genome, and we use a separate computer node to work on each fragment. So using massive parallelization. And then yes, we can deal with large data sets. That's not really fully satisfactory for those two reasons.
Oh, I mean, it sounds pretty good to me, actually. Okay, well, I think we'll wrap up here. I got to run, but thank you very much, Xavier. That was a lovely talk.
Thanks for having me. It was a pleasure.
And I guess, let's see. So like you say, Tim is next month.
I'm looking forward to it. Bye.